Good afternoon, Dr. Shah. Rupesh sir, good afternoon.
Good afternoon. Is Ankur.
Yes, sir. Dr. Ankur Sharma. Thank you so much, sir, for joining.
Thank you for the invitation.
Thank you so much. We'll start after 5 minutes, sir.
Okay,
Audience are
okay,
joining, sir.
okay, sure. I will wait.
Rohit, please start.
Okay, sir. So first of all, thank you so much, sir, for joining us and giving the time from your busy schedule. So, sir, I'm audible?
Yes,
Hello.
yes, you are perfectly audible. Yes.
So very welcome, Dr. Rupesh Singh, Dr. K. Rupesh Singh, and I welcome you to this session. And let me introduce Dr. Singh to all of you. Sir has completed his PhD from Plant Molecular Biology and Genetics Engineering Department from the Defense Institute of Bioenergy Research, Nainital, India. Sir has completed his first postdoc in January 2016 at the Department of Biology, University of Meio, Portugal. And during this tenure, Sir has guided three young researchers, one PhD and two master students. And at the time, Sir has responsible to design the experimental setup and ensuring the high value results. Sir has completed his second postdoc in March 2020 in the Department of Agronomy with Viniculture Lab University of Trás-os-Montes from Portugal. During this tenure, Sir has working as senior member in the lab and in the management of one research line in the innovation and wine. During this tenure, Sir has leading the team of 7 young researchers. After two postdoc projects, Sir has moved to Ireland to gain the experience in industrial research and started working as principal investigator in MBio. And major duties at the time, Sir has Sir leading the team of scientists, technician, and researcher trainees, project management, and establishment of new research facilities. Sir has 11 year of post PhD experience in diverse area of sustainable agriculture, horticulture, soil and environment sciences, molecular biology, food technology, plant pathology, genetics engineering, and modern genome editing technology. Sir has 2,631 citations and H index of 24 and 99 peer-reviewed published scientific communication, including one grant patent and one provisional submission recently. Sir is currently working as a researcher biologist in Sitab, and Sir has started a new technology research line for the better management of resources, mitigate the emerging contamination in the soil and compost, reduce greenhouse gas emissions, and soil nature properties restoration by natural-based solution. Sir has recently invited as an external expert by National Research and Development Agencies, Government of Chile for evaluation of research proposal for national funding. So very welcome, sir, to this session, and over to you, sir. Thank you so much, sir, for joining.
Thank you, Rohit. And first, I would like to say thanks to all the team and for this invitation to interact with the young researchers. And as this is a workshop, maybe I prefer to share my experience, not like a typical presentation, what I did in past. And as you already introduced that there were a lot of different research areas I was working. And whenever I see some new research line, I like to interact and incorporate my research with them. And that's how we develop a scientific curriculum. So let's start with some slides. Let me share.
Rohit ji, kindly start YouTube live.
Done, sir. Done.
How I can share the presentation?
There is a green button, sir, on the share button over there on your screen. Just click on that, and you can share your screen with us.
Yeah.
Yes, yes, yes.
Put in the presentation mode. Yeah. Now you can see the presentation.
Yes, sir. Yes, sir. It's visible.
So let's speak a little bit about how you transform your research, what you do in the lab, and when you finish this work, how you make it to a scientific communication. And basically, I will share my previous experiment, experience, how I manage, how I learn, and for me mainly it was a learning by. So let's start. This is the entry of our university, which is called the University of Trás-os-Montes and Alto Douro. In the back, you see some mountains. So this is the mountains of Trás-os-Montes. And these are the magnolia flowers now is blossoming in the university, as here it is starting now the summers. So let's start from the beginning. When you start working in the lab, the first day you go and what you think. So what I did is first I think to organize myself, organize the things, what I am going to do, what is the experiments I am going to start. And the basic in the research is to learn the best lab practices you can do, and those lab practices goes with you until the end of your career for always, and those are the most important to perform your research, to write your research paper, or to write your thesis, or to work with your colleagues, to work in a lab, how you make your reputation that you are a good researcher. Here, why I'm saying is because the misconception is if we work a lot, we are a good researcher. But is not like that. If you work a lot, you are not a good researcher if you are not organized in your work. And in the research, you think more and work less to avoid the circumstances that the same experiment you can do 5 times, you repeat, you generate a lot of data, but the same experiment you do at once, but with proper planning, with everything organized, and you get the better results than repeating it 5 times. And the first thing what you have to do, especially the young researchers, is to think what is the significance of your research. For example, when I started this postdoc here in University of Minho, the project I started is metabolite engineering in a plant is a Hypericum perfoliatum. So Hypericum perfoliatum has a lot of significance for pharmaceutical industries. It has some small glands in the leaf, and they produce a biocompound is hypericin, and it has been used in the cancer drugs, in antidepressant, and is a lot of other benefits. So when I start researching like the significance, and the significance one you think, okay, is a very demanding compound, is a very demanding plant, is a very good work. But what is the novelty? Like what your work will add to the existing research which has been already done. So you have to make a state of art. Here in Europe, this is the review literature. Here we call it state of the art. So you go from the historical background until the most advancement has been done in that area. So historical background, you have to study like the plant where it was originated, like the below centers of origin, how it evolved, why these glands has been evolved. Is there some hypothesis already studied? Or there were some modifications during the evolution of these glands in Hypericum perfoliatum, and you make a chart that all this thing has been done, and then you see some review literatures that what are the recommendations for the future research, why you should do this research. Because if you don't make it in 1 year, when you finish your experiment and you see that this already has been done and there is no novelty, and when you start writing your communication, this is the first comment that what is the novelty of your research, what is the significance of your research, why you perform this research, why you use all these resources if it was already studied. So this is the first and basic thing of your research when you start as a student, as a PhD student, or as a master's student, or as a postdoc, or even as a senior researcher. Whatever new research line you start, you have to make this study, this chart, to clarify your ideas and your future plan, that what you are going to perform and how you can transform these results into your scientific communication in future when you start writing the paper. Then second most important thing is the methods you use, because there are in the science, it has been evolved very significantly. There are always new methods, new technologies to analyze the same results. For example, if you take the microscopy, 20 years back, the microscopy was like very basic, but if you see now, there is very advanced microscopic is advances. So there are always some limitations, that maybe you don't have access to the advanced technologies. You have only the basic methods. So you try the basic methods, but meanwhile you try to find some collaborations or some facilities or some scholarships where you can travel to those institutions which develop these advanced technologies. You try to learn, you study the most advanced methods to improve your scientific skills. And then another important thing in the methods, if these methods are accepted by international scientific communities, because there are still some basic methods which are even if it's 50 years old method, they are very significant. They have potential to give you the good results, and they are accepted by international scientific communities. So choosing the method to perform your studies, you have to keep these two things in your mind. If is advanced methods which give you the precise results, or these are the basic methods, but it still is accepted by the international scientific community. And while performing your experiment, this is the very basic thing which I learned during my PhD in Indian Institute of Bioenergy Research, because I was working with molecular plant biology. And my main work was to identify the novel genes from Cicer microphyllum. Cicer microphyllum is a legume plant, but is a wild legume. It grows in the very high Himalayan mountains around 7,000 meters in Changphang Valley near Kargil. And you have very less samples because it grows only from May until July, August. Then is everything is dead, is covered by the snow, and the place is called as cold deserts. So if you don't collect enough sample in these 3 months, you don't have plant samples for rest of the year for your studies. And it's not possible also that every year, if I was working in Nainital, and from Nainital to go to Kargil to collect the samples is very hard task. So the sampling is very important in the research work. Okay, if you have normal working with normal plants like a tomato or capsicum or carrots or horticulture crops, you can grow them, you get the enough sample. But if you are working with some rare plant species or some species which are not easy to find in nature, you must be very careful with the samples. So what I did, I collect during those time when the plant samples are enough in those places. I collect in big amount, and then I make different parts. One, I freeze all the green samples as it is the whole. I put in -20 in plastic bags, and you make different partition. You don't put all the samples in one plastic bag. You make 5 or 6 small plastic bags. Put the date when you collected, from where you collected, if it's from the same place or if it's from the different places. You freeze in -20 or in -80 for long-term use. Second, I extracted the DNA by MaxiPrep method. You can extract in big amounts the DNA. You freeze it or you lyophilize it. and you make different aliquots and you store it in -80. To store the RNA is a bit difficult. So usually I store the green plant samples and the DNA, and which was very useful for when I finish my work. And I will discuss this part in the next slides. Then second thing, you must have a lab book, and it must be updated every day. When you reach to the lab, you put, okay, today is this date, the 4th of April, 2025. You have a plan you are going to do. You make everything recorded there. What the method you use, you record everything, every small thing. It's not for someone else, it's for yourself. Because after 1 year, you don't remember what you did. For example, you extract some DNA, you run a gel electrophoresis is the simple one. So you choose, okay, is 50 millivolt, you run this genomic DNA for half an hour. But when next time you are doing, you are in a hurry to go somewhere or you don't have enough time and you run it only for 20 minutes with higher voltage. So just write it there. So next time when you see the results, you know that, okay, this gel was run for 30 minutes, but the other gel, I run it for 20 minutes. So just record everything. There is no shame that, okay, today, because you were in hurry, you are going for a movie, you don't want to say to someone else because the boss get angry or get upset. You don't worry about that. You just record your method, what you work. And then the next thing, when usually we perform the experiments, we use 3 replicates. This is normal lab practice, but I always use 4 replicates. Why? Because when we reach in the end of the data analysis, there is always it happened with me. There is always one replicate, which is very different from the 3 others. So if you need to treat the data, you remove that one replicate and still you have 3 replicates. And I see some of my colleagues when I do my PhD, okay, you use 3 replicates, but at the end, okay, one is not good. They remove one, they keep 2, they just add one manual, the average of these 2, and they make the 3rd replicates. That is not a good practice. So it's better you keep 4 replicates, you curate the data, you keep the 3 good replicates, you can remove one. So your data is like a good data, good lab practice. The second thing, when you have some results like some gel pictures or some blotting pictures or whatever it is, take many pictures and store the picture in the as it is, like the original picture with full pixel size, the best resolution. Always keep this picture with the date when you capture this data, and then make a copy from this picture to the presentations, because when you go to submit an article, now they always ask the original picture where it is. So if you make changes in the original picture and you don't have the original copy, you may have problem in the end. So always keep the original picture as a record and then take it a copy from there to make your presentations or your posters or to go in a conference or whatever you want to do. But the original one must be saved somewhere. Then another thing is don't collect a lot of data without organizing it. From the beginning of your experiment, you create a Word file. Today you extracted the DNA. You have a notebook. You write all the steps how you extracted the DNA. Even if it's available protocol, you have a lab manual, but write in your copy the steps you are performing. Yes, Hamid, you can ask. Hello.
The doubt clearing session will be start after the lecture, sir. So you can continue, sir.
Yes.
We'll provide
Hey.
10 minutes for doubt clearing session after the completion of
Okay.
the lecture. You please proceed.
But if someone want to ask something is for us is normal life practice. We
Okay, sir.
allow the students to ask.
Okay, okay, sir. Thank you, sir. Thank you. Rohit ji, please take care of this.
So yeah,
Participants can directly one-to-one session with sir.
okay. So I continue.
Yes, sir, please.
Okay. Sorry, Hamid, we take your question in the end. Yeah, so I was saying that to organize your results, so make it from the beginning. Don't wait for the end that okay, one day when I finish all the experiments, then I will go to start writing my thesis or my research article or communication. From the first day you start, you make a Word file and mainly the results. You have a picture, you make it a original copy, save some in one folder. You make a copy of that to put in that Word file and keep that picture in a publishable format. That okay, if I send this to a communication, it will be like this. You put all the legend, what is that, all the details, the small material method, how you perform this experiment in a paragraph formation. So when you finish this experiment, you already have a draft of all your results done there. Only and all your material methods in details are written there. So now what you need is only to add your introduction, a summary, or we say is a abstract, and in the end is discussion and references. And while you do this Word file, you correlate and analyze the results. Correlate means, okay, you have these results. What does it explain to you? You also write little bit explanation. Okay, I run this gel. This band was of this size because I have this marker and this. I was studying this gene, which has like 1.4 kb, but I have a band of 1.3 kb. So where is 100 base pairs? You have to always keep on checking if it's from different species, the reports. So maybe in the other species is 1.4 kb, but in the species you are studying is little bit less. So all those details, while you are performing the experiments, you can keep on putting in the Word file. It make very easy in the end when you finish your experiment. And the next thing is the discussion of these results with your colleagues and the interpretation. In Europe, we have this practice that you make a team. You build a team with your colleagues. You always try to discuss with them. Our colleagues, they know for each other what they are doing, what is their objectives. They present to each other. Whenever they have some free time, they discuss about it. So someone do some experiment, he come to ask me, oh, Rupesh, I did this experiment, but I got these results. What do you think about it? And I give my opinion. Okay, maybe it's because it's a different species and the reports available is in another species. So and that is the science that you discover always the new things, you have new findings, new observations. So he or she think, okay, yeah, maybe this is a correct explanation. So you go immediately, you note down in that Word file that okay, it may be a reason. And this also helps you to make a team, and that is very important because science is like a teamwork. If you want to work alone, is not easy. It's better you work in a team so you can incorporate different areas of research in your work. You have better skills. You have better correlation skills that you can correlate different things from different areas. For example, my second postdoc, I worked in the grape wines. Grapevine is grapes. We say grapevine because the grape plant, which is used to make the wines, is called grapevine. And this place I work is called as a Douro Valley, is in this mountain region and is a UNESCO World Heritage Site for the grape wines. We have very old farms of grape wine, and they produce a port wine. So Porto is a city here is Porto, and is produced in Porto city. So because of that is called port wine and is a patent from Portugal. So I work in this area for the grape wine, and the objective was to develop a formulation which could improve the quality of the grapes. And in terms of quality, because these grapes contain the color, which is anthocyanin, is very dark color, and they give a lot of health benefits, and they give a very nice red hue color to the red Portuguese wines, and especially for this port wine. So one is to improve this content because it has a lot of antioxidant properties, and second is secondary metabolites, or we say is total phenolic compounds in the grapes, which also has a huge significance. The first thing is very important to the plant because if they have higher phenolic contents, they are more resistant to the diseases, more resistant to the climate change, they are more resistant to the abiotic and biotic stress, they are better in plant growth, they are better in the plant health. Second, all these biocompounds give a lot of health benefits to the wine. Wine is a final product, and there is a difference between alcohol and the wine because the wine is produced by fermentation. So during the fermentation, you extract all these the beneficial compounds, and of course the sugar of the grapefruits is converted into alcohol. So the wine has 10 to 13% of alcohol. It depends on the different wines, but is in Europe and American countries is one of the most important drink with the food, and it is a very important crop for European Union. So what I did, like I was a plant biologist. I didn't know how to make the wine, the vinification process. So you collaborate with the people who work with wines because you make a application, you make some method you apply in the plants by genetic engineering, or you do some other stuff. You know that okay, the is improved. You study the molecular biology, but you don't know how to make the wine, the final product, or the grape juice, and that is food technology. So my colleagues, some they were from food technology, so I started discussing with them, okay, how we can analyze these wines. So then they say, okay, you have the HPLC, you have GCMS, you have GCFID. So I start asking them if they can teach me. They start, yeah, when I do the experiment, you come to us and we explain to you the basics, how you prepare the samples, how you extract the volatile compounds, how you extract the non-volatile compounds from these wines. And like this, you learn slowly, and you make a team. And when you have like this, you get more studies, more data. And when you write your paper, you have a team, which is an author list. So when you have people from the different areas or from the different centers or from the different countries, it gives more value to your scientific communication. And it also improves your scientific skills as well as your interpersonal skills, how you interact with your colleagues, how you develop a scientific team, then how you can develop a good scientific proposal for funding. Rohit, just one thing, is still I have time or I need to finish quick?
No, sir, you have
We
sufficient
have time,
time, sir.
sir. We have time,
Yes,
sir.
sir, you have sufficient
Oh,
time, please.
okay. So after when you do all these things, you finish like one assignment of your studies, and then you go to start writing your scientific communication. And in the scientific communication, if you already did what I explained before, you have a Word file where you already have the results, you have the interpretation of the results, you have methods written. Now you have to write an introduction. So first you design a title that the best title is, it should be concise, and it shows the key message of your work, like what you did exactly. When you read the title, when it goes to editor or reviewer, they read the title and they know that what is story it may have. It should be attractive, and it should be different from the normal titles, like you can write, okay, molecular studies in the Sisar microfilm to study the abiotic stress genes. Now, if you write like this, is a very general title. They read, okay, is just a general paper. So maybe it get rejected from the editor desk. The what you can write, okay, you study, for example, you found a very specific gene. So you put that the name of the gene, which is the key of your communication, okay. Like like this DRIP factor was induced during abiotic stress in Sisar microfilm. So you make it very concise and specific. Then second thing I explained before, the authors list. If you have a diversified team from 2 or 3 different research centers or 2 or 3 different departments, it give more value because it shows that your work is more collaborative, is not monotonous. So these 2 things is very important when you have different author lists, different affiliations. It influence a lot and it gives the impact to your scientific communication. Now I present a case study of myself. When I was doing my PhD in Defense Institute of Bioenergy Research. So this is my first publication, was in Protoplasma Journal, and it was rejected 11 times. Like, you can take it, okay, like a shame. Okay, my paper was rejected 11 times. But I don't take it as a shame. Okay, it was rejected, but what I can do? I have to try again and again. I never know that knew how I should write an article, how I should communicate, or how I present my results, how I discuss my results, how I make a conclusion. So I wrote my paper, and most of the time the supervisors or the boss, they don't have time to see your communication. So you have to go by yourself. So I started submitting, rejection. There were always some comments, and from the beginning they give you very basic comments. Okay, the abstract doesn't show the key message of your communication, so you must improve the abstract. So in the first rejection, okay, the abstract is not good, so I improved the abstract. Then in the second rejection, oh, your introduction looks like a discussion. This part is more to discuss, is not the introduction. So one by one, you see the reviewers comment why they reject. There is no shame that it was rejected. There are some other issues also, like sometimes the journal has is not in the scope, or they already have a lot of publications submitted there to consider, so they reject. But you always see the reviewers comment and try to improve as much as you can. Try again, submit again. So 11th time when I submitted in Protoplasma, it was accepted very fast by 3 reviewers without any suggestions. So from there, I learned by myself that, okay, these are the problems. And slowly I started improving. And now we have good chances. Like when I submit a paper, most of the time is accepted or is in major revision. There are very few chances when it get rejected. So what I want to say to the young researchers, there is no shame to that your paper was rejected. It doesn't mean that your paper was not good or your work was not good. If you organize from the beginning, you have good results, you have all the data, all the methods written, everything recorded. You just need to develop it in a good communication style, and you try again. Hello. Just more 5 minutes. And so, and for the rejections, the major comments what were that, as I said, your title must be changed. Abstract should be concise to give the key message. The introduction look like a discussion. Material methods are not described in detail. This is very common comment in the rejection these days, because most of the researchers, what they do, they put a reference that, okay, the experiment was performed according to this, and they just put the reference of another paper, and that is not enough. As soon as the editor or reviewer, they see it goes to rejection. In some cases, okay, if the work is very good, they may ask you a major revision, but in most of the cases, it will be rejected. The results and discussion should be in a story formation, should be aligned. That what are your results and how you discuss. It must be like a storytelling. Why you have these results? What is the significance? What it can add to the future research? And then the most important is conclusion. One paragraph, very clear, very concise, with the key message and how it is going to help for the future audience. So if you manage to do this in a proper form, you have a lot of chances to your communication will be accepted fast. Then another major issue. As I said, I always store some research material. When you submit a research paper, in many times they ask you a small experiment that, okay, you have this PCR picture. Can you make a PCR with this only with this gene and with a marker? So if you have stored your genomic DNA, as soon as you have the reviewer's comment, you go there, you take DNA, you perform a new PCR, a new gel picture. In the next day, you are ready to answer this comment. But if you don't have a genomic DNA, in worst case, if you don't have the plant sample, you have to grow again the plant, you have to again extract the genomic DNA, you have to do a PCR. It takes a lot of time. So be careful. This is very important key of your research, that always store one part of sample in every step. Like, okay, you bring the plant samples, keep some plant samples. You extracted the DNA, keep some extra DNA in the freezer. You make some PCR analysis, even you can keep the half of the PCR product. You have 50 microliter, you use 10 microliter to analyze, label it, store it in the freezer with proper labeling, the date, keep it there, and record it in your notebook that what the samples you have in your freezer. So in the future, if there is some comment, okay, this picture is not clear, can you make another one? If you have a PCR product saved, you just need to run a gel. If you don't have, but you have DNA, you need to run one PCR. You don't have a genomic DNA, but you have the plant samples, you extract the DNA, you run a PCR, you run a gel. But imagine if you don't have a plant sample, how you are going to do. And in my case, if I don't have plant samples, I wait for 1 more year to go to the Changthang Valley in Himalaya to collect the plant samples. And I could quit, okay, this paper is not going to accept it. So this is very basic thing in the research. Then another comment is like, you have to repeat some small experiment that I said that picture quality was not good and sometimes the results were not consistent. So as I said in the beginning, once you do experiment for 30 minutes, but next time you are in the hurry, you just run the gel for 20 minutes. So you go back, okay, why is not consistent? And you know the answer. Okay, because first time I run for 30 minutes and second time was only 20 minutes. So that is the difference. You make another gel, run in the same conditions, run in the same voltage, you make a picture and you get consistency in your results and you are ready to answer the reviewer comments. And then the third is the most common comment throughout the rejection is like poor English. And that is not to worry about much. You always like discuss with your colleagues, because as for me, I took myself as a case study. I studied in a government college in a small town, so my English was never good. So I always, okay, there are some words you use now is the internet. There are many softwares for autocorrection or even some help from the journals. You can improve the English, but okay, it should be simple. It should be when someone read, should understand what you want to say. You don't need to write like very high quality English or like very special English like Sasi Tharoor. Don't think about that. Just write in a very simple words. It should be understandable for everyone. And even today, I received this sometimes this comment that manuscript must be revised by a native speaker. And many times we give to a colleague, he read the paper, okay, is okay, and you resubmit it, okay, it has been checked by a native speaker. So these comments, don't take it very like heavy, you can manage it. But the previous ones, here, if you don't have your samples stored, those can be a problem. So this, you have to be careful from the beginning when you start your experiment. So organization of your research work from the beginning is very important. And then the ethical guidelines in your communication. There are some international committees, they provide the ethics, what you have to follow, like the Committee on Publication Ethics, scope, Committee on Publication Ethics, and the International Council of Medical Journal Editors. This is for medical science, because there are involvement of animals, and you have some specific guidelines. But for the plant science, there are not those things. So the US Office of Research Integrity. So there are different international societies, a lot of different guidelines for the ethics you have to follow. But you just need to know the basic ethics when you prepare your scientific communication. And the basic ethics for me is the most common is plagiarism. That plagiarism is like you take idea from someone else, or you copy some text from someone else. There is a self-plagiarism, that you have one paper published before, but next time you do the same thing just with a different species. So you just copy all the things there, you replace the species name, and you submit to the journal. That is self-plagiarism, and is also considered as non-ethical. So then, like you copy the text and change some words, but the citations are same in the same pattern. That also considered as a non-ethical practice in your scientific communication. Then another thing is authorship and contribution. This is most important in your research communication, because many times you ask some help for some experiment. Okay, you collaborate with someone. Can you do this experiment for me? The person, he does, and is a major part of your communication. But when you submit the article, you don't put his contribution. So if it's a significant contribution, you offer him the authorship. If it's a very less, you put him in acknowledgement. So this is the most important ethical issues in your scientific communication. And then the funding source, from where your work has been supported, from which project, or is from the center, or is from the university, who been used to perform your studies. You must put in your communication. So this, for me, these are the basic ethics when you prepare your scientific communication. First, you acknowledge all the people who participated in your communication, directly or indirectly. If they participated directly, significantly, they must receive a co-authorship. If it's not significant, but still there was some help, you put them in acknowledgement. You put all the funding resources in your communication. Don't copy from the text from the other articles, or even from your previously published papers. You make your own, like you publish something, but you think, okay, this is very significant for this. You make like a story, maybe with updated citations, but you change. You cannot just copy everything from the previous ones. And for me, this much ethics is enough to prepare your scientific communication. This one, I just put a disclaimer. Usually we use in the art, and it was very funny when I finished my PhD. My supervisor didn't agree to sign. I don't know why, and he said that you should put a disclaimer that all the characters and events in this thesis are fictitious, because I don't see you working in the night, late night hours, in the weekends. So I always keep this disclaimer, and I always say, okay, we must have a disclaimer in science that all the characters and the experiments in this thesis or in this communication are true, and they have resemblance with the events, with the people. Is none of a coincidence exist. So what I present in this communication is 100% true of my belief. So that's all the my experience and what I learned, and I wanted to share with the audience, with the students. And now I'm open for to take any questions, suggestion from the audience. Please go ahead. Vishnu is here. Hello, Vishnu. Thank you so
Yeah,
much for this
yeah, Dr. Saab,
invitation.
thank you very much for wonderful session. And I really, really appreciate that you talked a lot about publication ethics and ethics, scientific ethics, and especially the last one, like we need to put disclaimer in scientific publication. I think it is needed to, I think it's good for the thesis, but I think it should be in paper also, because now you see the large number of scientific publications are getting retracted. Many journals, even in many of my colleagues, getting like that, sometime without any big issue. But of course, I mean, scientific malfunctioning, malpractices, and this fabrication of data, plagiarism. I mean, one work can be published. I mean, authors are publishing in two places, in two journals. Even one experiment they are segmenting in different scientific articles. So really appreciable lecture, especially to young researchers, to know what ethics they need to follow, and they need to be careful before submitting their scientific manuscript. So thank you, Dr. Saab. I do not have any query, because it's really great lecture, great talk based on your experience, and very thankful. Здравствуйте.
Thank you, Vishnu. And I didn't present like a typical presentation. I just wanted to discuss my own experiences and how I learn and I improve. Okay, I'm not a
I
big scientist, but I learned the normal practices, and I thought is important to
Dr.
discuss with the
I think,
students.
Dr. Saab, your experience is really necessary here to talk, because this whole 15 days workshop devoted for the scientific writing. So, of course, the talking the same thing shouldn't, I mean, be a good idea. So all three presentation I listened, like yesterday Professor Wong presented, today you, and day before yesterday I was. All three lectures are completely different and based on the personal experience. And you are the great, because you really aided many additional things in these scientific publications, scientific writing. Maybe Dr. Nandani or other colleague, Dr. Ankur, wanted to say something about Professor Rupe's lecture, and maybe some audience have query. Maybe we can take some. So thank you, Dr. Saab.
Thank
Thank
you.
you. Many
Any
thanks.
participant has any query, may ask. Open is yours, please. Chat box is also open, and audio box is also open. Any participants has any query, please ask.
Good afternoon, sir.
Good
Good
afternoon.
afternoon, please.
So I have one question. Like we perform several experiments. If we publish paper with few experiments, can we include those experiments in another paper, which includes all the experiments?
Like the same experiment you want to include in another publication?
Yeah, for example, sir, if I am working with certain microbes for checking their antagonism and plant growth promotion activity, so can I publish the paper, single paper with only antagonism results, and another paper with TGP activities as well as antagonism of that microbes?
Like another paper you can publish with the plant growth promotion properties, and you can discuss your antagonism mode of action, which you already published in the next paper. You can give your own reference that in previous paper we publish this and these what the finding. And in this publication, we present the plant growth promotion, and then in the discussion, you can make a joint story how you discuss with the new results with your previous results. But the same experiment you cannot put in the material method and the results, because that is also a kind of plagiarism that this result has already been published.
Okay, thank you, sir. Thank you.
Thank you. And I would like to add one thing. Vishnu, you can please share my email if anyone want to ask something. They can write me an email after, because
Yeah,
I
sure,
think
sure.
Definitely, sir. This will be a great way, I think.
is rupesh@uyutat.pt is very. Vishnu has my email.
Yeah, I will share. I will share here.
Hello,
Yes.
Dr. Rupesh. Very nice presentation, Dr. Rupesh.
Thank you.
I have just one question regarding your presentation. One of my junior asked me, you give the suggestion that instead of taking 3 replication, we should try 4 replication.
Yeah, because
Yeah.
sometimes
Yeah.
sometimes you finish your question.
Yeah, yes, he told me that he's saying that because instead of taking 4 application, can we take 10 replication and choose 3 good replication results in our paper, so that our result will be going to far better than 4 applications.
No, I think
Then
then
I told him, told him that it's become fraudulent because you are playing with your results. So
yeah.
don't try to make things much more better instead of removing your wrong replications to get a fruitful result. Is
The
it
best
okay or
no, the
no?
best
I want to know
strategy
your opinion, bus.
the best strategy in this, like if you want to make 10 replications, so you make 10 technical replicates, you divide, okay, like you have one sample, you make 3 biological replicates, then from those 3 biological replicates, you make 3 technical replicate from each. So in total, you have 9 replicates from one sample, but those 3 are technical, represent to 1 biological replicate. And then you have 3 biological replicates out of 9. So that give you a very good data. But if you do like 10 replicates and you choose 3, that is not a good strategy.
Right. Absolutely right, sir. I also agree with you, and also he's listening your voice also. But I think you are absolutely right. Yeah.
Because in biochemistry we do a lot. Like each sample we divide in 3 biological replicates, and then each biological replicates have technical replicates, because, for example, the gas chromatography is very sensitive. So if you don't use technical replicates, you give a very big variations.
Okay. Thank you, Rupesh.
Thank you, Kshitij. Thank you.
Next, please.
Sir, we have queries in the chat box, and like lots of queries actually, and lots of appreciation are there for the presentation was good. And one of
People are saying, sir, you are very humble and polite person, very genuine person. People are
Oh.
writing such type of appreciations.
Yeah, now I'm reading. Thank you so much. The audience is very amazing. They are very humble also. And this is also like we learn in the science, because as much as you learn, you become more humble. So is a sign that you are learning.
sir, there is a one query from Anit, and the question is, sir, I didn't have enough paper writing experience, and being a beginner, how can I start my research writing, or what are the factors I should follow?
Like in the beginning, I said that you must be very organized. Like from the beginning, you have a a good methodology. You discuss, like, I also didn't have any experience, but I discuss with my supervisor, with the colleagues, with the literature, that what methods I should use. And you make a state of art. So you make a like experimental plan that what you want to do. And then you perform all the experiments, and you put all the results together. You make a very good explanation of your results. You make a detailed material methods, how did you perform, and then you discuss with most recent publications or most recent findings, and you make a story out of your publication that you did this work. This was the needed. The significance is in the introduction. Then material methods are completely in details, that if the reader read the material method, they know how they can perform this experiment. Then results you explain very well. You discuss it in very like in a storytelling form. Then a nice conclusion with a key message. And that is the key of your scientific communication.
Okay, thank you, sir. And then next one is Prajakta, I think. And the question is, sir, kindly suggest some software for reference management, like how we can use Mendeley or
Like
those. Yeah.
for reference management, we usually I don't use software, but I forget the name.
Sir, one is like Mendeley and like Zotero. I use those software for references
Is
generally.
good is good like with the software, but it's still you have to check one by one all the reference. That is
Yeah, yes,
most
yes, sir. It's
most
classic way.
tedious. Yeah.
Yeah. And one more question we take, like how to find, like the question is, which is the best journal for genetic engineering? But I think I can modify the question at how to find the best journals according to your stream or like, study.
Now there is some new services by the publishers, like I see in Springer and elsewhere. You can put the keywords and the title of your publication, and they suggest you a list of journals. They give you like I think 6 or 7 journals, that which one is the best in according to your work. But this comes from the very beginning of when you start organizing. When you have a work, you start searching your work. That okay, where more resemble work has been published, and like what I did. So I used to search the research articles, and I always pay attention about the journals. So I made a list of 10 journals in the first year of my PhD. That okay, I have to. These are the most important journals of my area, because that time we didn't have this much internet and like a lot of open access journals. So that was my strategy. But these days there are very new things. Like I said, in these journals, you can put the keywords, and you already have a list that where you can publish.
Thank you so much, sir, for the answer. And we have like question from Manpreet, and the question is how to improve scientific writing, like slash poor English. And how to improve
So
scientific writing, sir? The question is.
to improve the scientific writing, basic is reading. As much as the research articles you read, you improve like normally. You don't need to think anything. You will improve your English. You will improve your ideas. You will improve your correlation of the results, how you correlate from one result to another, how you discuss. So the basic is reading that I recommend.
Okay, thank you, sir. Then we'll take the last question of the session, I think. And think the question is by Dr. Preeti, and the question is, what should be the important points we have to look for selecting research journals? Because nowadays lots of like predatory journals are there. So how we can identify or go for good journals?
To identify, like we always choose the Q1 journal, like you have the Q1, Q2, Q3. Itself, they say like why is a quality one journal. So basically we go Q1 or Q2. Then quality 3 journals, you already know that, okay, they may be good. They can jump next year to quartile 2nd or quartile 1 category. And if you try always in Q1 journals, even if it has low impact factor. Like I usually don't go for impact factor. I usually choose, okay, is quality 1 category. For me is enough, is indexed in Scopus, indexed in Web of Science. Even if it has low impact, it doesn't matter.
Thank you, sir. So, Ankur, sir.
Thank you, sir. Thank you so much for joining. People and participants are saying you are very humble and polite. Really, sir, you are very humble and polite. And thank you for
Okay.
accepting my request to deliver a lecture. And special thanks goes to Dr. Vishnu, sir, for sparing time. Into yesterday's lecture, you were there. Thank you so much, sir, for coming and joining and supporting me. And thank you so much, Rupesh, sir, and looking forward for your support in the future activities organized by our foundation and university. Thank you, sir. Thank you. Thank you so
Yeah,
much.
sure. Please let me know in in 1 week advance, and if I can participate is always a pleasure for me. And I thank all of you, Ankur, Rohit, all the audience, and Vishnu also, who brought this opportunity. So
Thank you
thank
very
you so
much.
much. Thank you, all of you. Thank you, Vishnu, then.
Thank you. Okay, Dr.
Okay,
Saab,
then.
thank you.
Okay,
Bye.
okay.
Thank you so much. And
Thank you
all the
so much,
good luck
sir.
to the new researchers. And anything, if you need from me, you write an email, and I will try to
Yes, sir. I
answer.
already provided, sir. Already provided, sir.
Okay. Thank you so much then.
Thank you, sir. Okay.
Thank
All
you, sir.
participants can leave. Thank you. Bye-bye.